This is our presentation for our machine learning project. Our project, we are creating a model to auto count some fluorescent protein E. coli bacteria. So the problem here is that the manual counting of E. coli on a hemocytometer slide can be slow, subjective, and very difficult depending on your microscope and the size of the cell and the different slides that we have and the imperfections present there. And being able to count these E. coli cells are important for a couple different things. Bacteria counting and quantifying that is very important in industry, and for us specifically, we have an undergraduate laboratory activity in which the students will grow and count their cells to look at different concentrations of oxygen and other things that might affect that growth. And so the objective here is to create an automated computer vision counting process where we will, one, process the images, and then two, pass it to a model which will then give an accurate cell count is our objective. The challenge here is being able to train the model to differentiate between cells and visual noise, such as glass imperfection. Down below we see a little example of the pipeline of from the left to the right, what a raw image someone might see when looking through the microscope, and then how we're going to crop that and process the image to then train the model to identify what is a cell and what is not. So going into that with the pre-processing and the preparing of the images for our model, first off with our pre-processing, we have kind of two steps. First, an auto cropping step. We see that in the top right image where going from the initial bordered image, we have developed an algorithm to automatically crop to create 4 separate cells, so to speak, of the image from the one hemocytometer slide image. Then going below in that image, we then process that to try and subdue the glass imperfections based on their color and intensity, and amplify just the bacteria. And we've had varying degrees of success. The main issue we run into is that while we can accurately subdue or erase a glass imperfection, that those glass imperfections will overlap with bacteria from time to time, and then you end up erasing bacteria as well, which we do not want. And so we've had limited success to find a balance between there. So then, in going into preparing these images that we processed for the model, we manually annotate the images to create masks, which then are used to train and to test the model. Initially, we started with a cell pose Cyto3 model and have transitioned since then to a YOLO instance segmentation model. And there were a couple of reasons for this. Mainly, cell pose is optimized for high cell densities. If you look at the image in the bottom left, you see touching adjacent high density cells, which cell posed is ideal for counting. However, ours are sparse and they are usually, if they're touching, they're in long strands, but not throughout the whole image. And so the custom drawing that we can do in segmentation through YOLO, and we did that in RoboFlow, was much better. So once we've created those masks and manually annotated our processed image, we then trained our model on it. And this is what our model is at right now. So we tested it with a few YOLO instances, and YOLO 11S-seg 1 out was the best. However, none of them performed super well. In terms of just looking at false positives, it didn't do too bad, but in general, looking overall, it was not a very precise model. And so we're looking at different ways to improve this, including higher image quality and resolution, as well as better image processing